second Kingsbury Lecturer of the Year, and it is my real pleasure to introduce Ramon Parsons, who is the director of the Tisch Cancer Institute and Dean for Cancer Research at Mount Sinai. Dr. Parsons has been in this role as director of the Cancer Center since 2017. And they recently got NCI comprehensive status, which is, of course, very good. Better than very good, outstanding. Prior to being at Mount Sinai, Ramon was for many years at Columbia in the departments of pathology and medicine, and ultimately was named the Avon Foundation Chair for Breast Cancer Research. He is a member of the National Academy of Medicine and the American College of Physicians and the American Society of Clinical Investigation. And we were just talking about the fact that when he was chair for two terms of the AACR Special Conference Committee, he actually initiated and planned 75 scientific meetings, which I think is pretty impressive. Of course, most notably, Ramon is known for identifying PTEN, a tumor suppressor gene that, of course, is in very much at play in many cancers, including breast cancer, brain cancers, prostate cancer, and endometrial cancer. And weren't you part of that Stand Up to Cancer grant years ago that Ian and I were both part of? Yeah. And I had the pleasure of working with Ramon, I thought so, in this Stand Up to Cancer grant now probably a decade or more ago. It was a long, long time ago. Without further ado, Ramon, thanks so much for being here. And he will speak today about improving tools for targeting AKT in cancer.
Thank you, Eric, and thank you to all the faculty here for welcoming me. It's really a special time to get a chance to actually in person visit another institution and see old friends and familiar faces and catch up. One of the things that's really remarkable about that Stand Up to Cancer grant that Eric and I had the privilege, and others here actually, I believe was part of that grant also, was that at that time we were super excited that the PI3 kinase pathway was going to be a really important pathway for treating cancer. And it's been a pretty sobering ride. And it just shows you how when you draw something up on the drawing board, it doesn't necessarily work out the way you expected. But I'm kind of a stubborn person, and I just like to hammer the same nail over and over again. So I'm going to be talking about, ways we can try to improve targeting AKT in cancer. And I think the big problem, there's several issues, but I think the largest problem and barrier for targeting AKT is the therapeutic window. AKT is involved in so many different physiologic processes that our body needs to accomplish in its day-to-day function, that it's quite challenging to inhibit AKT without having a side effect that is consequential. And I'm not going to go into the details, but there's been several attempts to inhibit AKT, and I'll talk a little bit about capivasertib later. But it's pretty clear that there's some substantial side effects, including GI toxicity, hyperglycemia, so basically a metabolic toxicity, and also very severe rashes that can occur. And these are on-target toxicities because you see them with all the different AKT compounds that have been developed over the years. But what the excitement that, was really began in the early 2000s at this point, so 20 years ago, really started with the initial discovery of the AKT as a retroviral oncogene in 1977. Took 10 years to clone the human homologs. It just shows you how slowly things went back. I just phenomenal. And the same Stephen Stahl was the first author on both papers. Basically, he started this field, and then in the 80s and 90s, it was discovered that receptor tyrosine kinases, PI3 kinase, P10, RAS, PDK1, and obviously AKT itself. Even some of these discoveries, even in the early 2000s and mid 2000s, were genetically altered either by copy number change or by mutations that either inactivated P10 or activated RAS, PDK1. So basically, you have this really nice pathway where mutations in RTK, KRAS, PI3 kinase, P10, PDK1 can upregulate AKT in addition to mutations in AKT itself. And AKT, as you can see here in this little cartoon, regulates a host of different signaling pathways, with I'd say a very important output being the mTOR pathway down here, the TORC1 complex. And then over 10 years ago now, Gady Getz's lab basically did a comprehensive analysis of mutations across over 20 different kinds of cancer. And then this is just a plot from that paper, which looks at the Q value of combining a pan-cancer. Can you see? Yeah, you can see my cursor group here, combining all the cancers versus each the significance within any individual cancer type. And in terms of mutations that affect activation of the PI3 kinase pathway, I just highlighted them here. This is a box pull out of this group over here, where you can see some of the most frequently mutated genes we know activate PI3 kinase. They, EGFR mutations also activate the MAP kinase pathway as well. But and you also see mTOR here and FGFR at lower frequency. So it's a pretty sizable problem. The fact that we really don't have effective targeting of this arm of the pathway like we do for the MAP kinase pathway, which isn't even all that effective either, by the way. But so one of the things that that it's really important to think about as we talk about AKT is where can we capture a therapeutic window better than we currently can today? And this is a paper. This is a model taken from a paper published in 2018 in PNAS that's based on structural data, variety of different types of data combining hydrogen deuterium exchange, crystal structures, and other types of assays that basically have direct evidence and also inferential evidence for this cycle occurring in terms of the regulation of AKT. And here you can see in the inactive state something that's really quite interesting. It's sort of hard to appreciate in this model, but basically the pH domain, which interfaces with PIP3 normally up here, is actually locked into the kinase domain. So they basically interfere with each other's function and block activity. And through basically an equilibrium here that favors this construct, occasionally you find a release of the PIP3 pocket from the kinase domain, which allows for the protein to sample the plasma membrane for the presence of PIP3. And if it encounters PIP3, the protein is stabilized on the plasma membrane, and you see a very marked change in the structure of the protein from this form here, A, here to C. And this allows for AKT to be phosphorylated by PDK1 and then mTORC2. And those phosphorylation events, in addition to the release of the binding to the kinase domain, allows for a shape change in the catalytic cleft that allows for engagement of ATP and also after phosphorylation, and also engagement of substrate. Once the substrate is phosphorylated, it's not really clear how many times this can happen before, but basically once PIP3 is removed, from the signal. The autoinhibition occurs. This folding back occurs. You change the shape and structure of the protein so the phosphorylation sites are exposed to phosphatases, which dephosphorylate the protein here and here, and then you get back to square one. So, in principle, it would be nice to be able to inactivate the active form of AKT. And however, if, in normal signals that are physiological, if you have a level of kinase inhibition that is allowing you to inhibit the signal in the tumor, it's also going to be at a level where it could inhibit the signal, in that occurs normally under physiologic settings in let's say in endocrine signaling, for instance, in insulin signaling in our bodies. So the various inhibitors that have been generated to target AKT, here's just a list of some of them. Some of them, like MK2206, are allosteric inhibitors, and they bind to AKT in that inactive conformation. They lock it in that inactive conformation. And others like AZD5363, which led to the development of capivasertib, is the same name. It's the original name for capivasertib, is an ATP competitive inhibitor. And there are several other inhibitors that were developed that never made it finally into the clinic, so far haven't made it into the clinic. What's interesting, as I alluded to, is that the allosteric inhibitors lock the protein in this inhibited state when it's not phosphorylated. And then here in the GDC, which is an example of an ATP competitive inhibitor, it keeps it. It's actually in the active state when it's phosphorylated and the pH domain is open. So it's it's from all the work on the ATP competitive inhibitors. They showed a therapeutic index, basically preferentially inhibiting the growth of cells that had activated AKT versus ones that didn't by about one order of magnitude. Like that was documented in this paper out of the Genentech from the Lin lab at Genentech. So anyway, back in November of 2023, capivasertib was approved to use in combination with fulvestrant in hormone receptor positive HER2 negative locally advanced or metastatic breast cancer. It was also approved in Japan and the EU. And it was the first FDA-approved inhibitor, but about 13% of patients have to stop taking the drug because of toxicity issues. And certainly the dose, there's many patients that have mutations that don't respond. It's certainly not a perfect drug by any means, despite its demonstration of improved efficacy over fulvestrant alone. So anyway, actually, starting over like 6 or 7 years ago, I started collaborating with John Jin, who's a medicinal chemist at Mount Sinai. And we decided to see if we could try to make degraders that could degrade AKT when it's in its activated form, using with the knowledge that the ATP binders are more potent, are more have the slight advantage, about a log-fold advantage over in terms of growth inhibition. And so we started down this, I'm bringing coals to Newcastle here. Obviously, this is the home, Craig Crews, with the invention of the PROTAC. So I don't think I need to spend a lot of time on this. This is a transformative technology that is, I think, likely to lead to actually FDA-approved agents very soon, if not already. But there's been some very nice data for like ER degraders, for instance, that's been published. But anyway, so we commenced an iterative workflow where we started receiving molecules designed by the Gin Lab for us to measure if they could degrade. And we basically had an assay using Western blot to measure degradation. And then we also, those that were able to degrade, we sent out to a CRO to test for oral bioavailability. and just not and just bioavailability in general. And then we optimize structure, and we did this. We did this on about 8 iterations, about 200 molecules, led to about to 4 different papers that we published in a range of different degraders using either VHL binders or cereblon binders and a variety of different AKT binders, whether they be allosteric or ATP competitive. And we compared them across many different cell lines, in some cases up to like 38 cell lines, in other cases smaller numbers of cancer cell lines, for their ability to not only degrade, but also inhibit growth. And in some cases, we did look for efficacy in mouse tumor growth assays. So anyway, this is just one of our lead, compounds that we published, which is basically an analog of the AZD drug, which is then tethered to a VHL binder. In this case, MS-21. I'll be referring to it quite a bit in the next few slides. So MS-21 is a VHL-based ATP competitive binding degrader of AKT, and this just shows you the binding efficacy of the degrader versus binding to AZD itself. And you can see you take a hit by adding the molecule, more so with AKT2 than AKT1. And in many cell lines actually do also, which is AKT3 is the isoform that's principally normally in the brain, but cancers often acquire expression of this as well. So anyway, so the models that we chose to optimize degradation was actually we stumbled upon it basically after trial and error, but the PC3 prostate cancer model, because it's a PTEN null model, and we were seeing very, and we wanted to screen on degradation based on a system where we know AKT is really active and most of the time, and that prostate cancer in general has very high AKT activity signal compared to even other kinds of cancer. And we, for not clearly understood reasons, actually, but in any event, we identified this MS-21 was able to degrade AKT at this, low nanomolar concentrations, and this is just a time course showing that that the degradation of AKT took actually, at least about roughly 4 hours to get to about half degradation, and it took more like 24 hours to start to see really striking downregulation of downstream signaling, which is quite distinct from what you see with a kinase inhibitor, where you see very rapid downregulation, but then feedback activation of the pathway by 24 hours. And this is something also Alex Toker's group, actually, who it turns out is talking here today, had a similar story using the degrader they developed, which was based on the GDC 0068, the Genentech ATP competitive inhibitor. This is just showing that these are controls for making sure the PROTAC is functioning as expected. I'm not going to go into the details. And this is just data showing what we were saying, comparing AstraZeneca's drug in terms of colony formation. You can see an improved inhibition in colony assays for these 2 different cancer cell lines that are missing PTEN. And you can also see the improved cell death that we were seeing compared to the AKT inhibitor, AZD5363, which is actually, we're still not quite understand. This is just a cell death marker. We don't understand what mechanism of death or combination of mechanisms of death that are occurring here. So proteomic analysis of the degrader, obviously we wanted to see if we were hitting the targets. And so because the mass spec is not a perfect, proteomic mass spec is not perfect, we could, several times we could see AKT1 and 3 degraded, AKT1 and 2 degraded. So we wanted to just keep doing the experiment so we could document all 3 peptides for all 3 proteins were flying in this, looking at a ratio between MS-21 and an inactive molecule. And we, in some of the experiments, not shown here, we could see clearly aurora kinase B was being depleted, and also PDCD4, which is a known downstream a substrate of AKT and regulated by AKT phosphorylation. So this is like an internal positive control. Here you can see this is just looking at aurora kinase B. We're seeing very nice downregulation of aurora kinase B with the degrader, better than we were seeing with the AZD inhibitor at 24 and 48, 72, and 96 hours. And we also saw better downregulation of a AKT substrate. It's subtle, but it was reproducible for the MS-21 compared to AZD. You can see that here at 72, 96 hours. You can see that really doesn't show up until 96 hours here with the 468s. Actually 72, you see a little bit of it. Aurora kinase B is known to regulate mitosis. And so when we looked at the profile of the cells, we were seeing hyperdiploid cells after treatment with MS-21. And here you can see the actual quantitation of the different phases of the cell cycle. Here you can see that the G2M is expanding here quite nicely. And this is just more signaling data showing that you see various amounts of downregulation of aurora kinase B, but the best levels were seen with MS-21 and also a very broad inhibitor to the PI3 kinase class 1 family members with GDC 0941 gave us the best downregulation. We then were able to establish that we have a predicted phosphorylation site for AKT and aurora kinase B. We then demonstrate, I won't go show the data in detail, but we could demonstrate in vitro that aurora kinase B could be phosphorylated as a substrate by AKT. And then we did some in vivo experiments demonstrating that aurora kinase B, when we made a mutation that blocked the predicted phosphorylation site and the autophosphorylation site, we could demonstrate reduced phosphorylation of aurora kinase B using a AKT substrate antibody. And we similarly saw that we could inhibit that signal when we inhibited PI3 kinase, as you'd expect, or AZD, which is the AKT inhibitor itself. We also could make mutants that mimicked aurora kinase phosphorylation at the AKT site, and this led to stabilization of the protein, shown here. And this mimic and also just overexpression of the wild-type protein itself was able to partially rescue the colony formation data. So showing that aurora kinase B downregulation could be a valid downstream biomarker for efficacious anti-tumor growth effective. I mean, it's known, for instance, that inhibition of aurora kinase B causes cell death due to catastrophic mitoses. So, and this is just more rescue data in different cell lines, showing that it was reproducible in different cell lines. So we were able to get reasonable doses of this drug after with IP dosing. You can see the concentrations here over a 12-hour period of time compared to AstraZeneca. And we also saw very nice tumor growth control here compared with 2 different doses of the AstraZeneca compound in the PC3 model. And this is also in the MB468, which is a triple-negative breast cancer model. The other thing we did is we wanted to, look at the tumors themselves to see how they were responding. And it was pretty clear that you can see that we could affect the level of AKT in the tumors you'd expect from the degrader. We could see sort of comparable downregulation of phospho-S6 phosphorylation with both, but better inhibition of Ki67 with MS-21 quite clearly. And also the other issue about this compound was, despite the relatively high concentration in the plasma, the tumor concentration was about a log-fold lower, really kind of at the border of, we were kind of lucky, frankly, to be able to, if it had been another log lower, it probably wouldn't have worked very well. So anyway, we we then profiled 38 cell lines of various genotypes to see how they compared to treatment with the AstraZeneca compound, AZD5363. And we basically found, we classified cell lines into resistant to MS-21 versus sensitive. And what we found is that we were able to convert quite a few cell lines from resistant to sensitive with the degrader versus the AKT kinase inhibitor. And in general, when we looked at this resistance profile with the genotypes, we could see clearly enrichment of sensitivity, which is in blue, when we had a mutation of the PI3 kinase P10 AKT pathway. This included HER2 amplifications, by the way. And however, we also saw that if we excluded the RAS pathway, we saw better evidence. So basically, these are mutated without RAS mutation. We could see better sensitivity versus resistance. And then also, if the pathway was mutated, we only had one example of a BRAF. Actually, it was a non-canonical BRAF mutation in one cell line that was sensitive. All the rest of the BRAF and KRAS mutant cell lines were resistant. On this side, we also had obviously, if everything was wild type, that was the spectrum there. And just to show you, this is just mapping those 8 cell lines that are converted from resistance to sensitive from the capivasertib or AZD5363 to MS-21. You can see them all shown here. They all are lines that have PIK3CA or P10 or HER2 or AKT alterations. How are we doing with time? So you can see that here's PC3. We can degrade all 3 isoforms of AKT. Here's 2 cell lines that are quite sensitive and in blue that show nice degradation. And then we started to look at why are these cell lines resistant? Well, one thing that correlated quite well is the RAS mutated lines and even some of the PIK3CA RAS mutated lines. And here's P10 and BRAF mutated line. There's failure to degrade in the presence of MS-21 of the AKT kinase. And we still don't completely understand the reason for that. We can also, here you can see sensitivity also correlates with downregulation of aurora kinase B. You can see this cell line here, 468, which is actually, I don't show it here, but we found that its efficiency of degradation was not that great. And it turned out had very low expression of VHL1. So we overexpressed VHL1. We could see much better downstream signaling effects and degradation of AKT. So this may just be, but it's quite sensitive to even partial downregulation of the pathways. That's why we've seen it in the sensitive group. But you can see these resistant lines, they don't budge the aurora kinase B levels. And this is just showing you data using GDC 0941, a PI3 kinase inhibitor, broad spectrum, and showing that similarly has no effect on aurora kinase B, but it does affect aurora kinase B in most cases, except for this, for some reason, this cell line here in response to the PI3 kinase inhibitor. And so basically what we were saying, also just to point out, is just this correlation between AKT phosphorylation and sensitivity. And these are the resistant lines. And this is just taking RPPA data from the cell line encyclopedia. We just took the cell lines that overlapped. And again, we saw this nice correlation between sensitivity and baseline high levels of phosphorylation of AKT. With the exception of these 2 lines here, it turns out these 2 lines are interesting in the sense that although this is what we reported after subsequent looking into it and other papers that have come out, one of these lines has a mTOR fusion gene, And another one has a biallelic mutations on TSC2 and NF1. So not clear, so potentially explaining their resistance. Anyway, so really, I think a really enlightening moment for us, was when we decided, hey, can we just toggle degradation in resistant cell lines that basically are BRAF or KRAS mutated that were showing resistance to degradation? Is there a way for us to stimulate degradation in these cells? And what we found is yes, by simply adding IGF-1, and then we could now take resistance to degradation and convert it to sensitivity to degradation. And this is just an hour after stimulation. So it happens pretty quickly. And we could see this also in this pancreatic cell line with a KRAS mutation. And we also saw the same thing in these two cell lines when we stimulated with insulin. And as you know, insulin and IGF-1 are somewhat promiscuous in their ability to interact with both families of receptors. But clearly showing that we can, the activation state of AKT can lead to improved degradation in a cell line. And then, this is just a bit of a digression, but we were able to see very, very exquisite sensitivity to combination treatment with trametinib to inhibit MEK in combination with MS-21. Not in all instances. Some cell lines remained resistant. Some KRAS mutant cell lines remained resistant to the combination. So these are some of the best examples. But something like half of the time, we could see this restoration of sensitivity that was really quite impressive, in my opinion. Anyway, so this is work from several years ago now. And so we've literally been spending the last 3 years trying to figure out, is it possible to make something that is more useful clinically? So we were lucky to get funding from Henry Kravitz's foundation to develop these more of a clinical grade molecule. And we just continued to the same iterative workflow, this time absolutely looking at oral bioavailability versus just any IV bioavailability. And we've actually, at this point, this is about a couple months old now, this slide, but we've done 22 batches of compounds and a bunch of different series of different AZD series and GDC and GSK and modified series. And so at this point, over 500 compounds have been screened, and the promising ones have then been set off for PK analysis. At that time, about 69. Well, at this point, it's probably about 75 that have been sent out for testing. And we've identified some that are over 30% orally bioavailable. And the first compound sort of that, by the way, this was really tedious and really painstaking and did not work for like over a year and a half. And then after banging our head against the wall, we sort of made a breakthrough with this molecule here where we were seeing good, not great, but this was kind of our lead that led to subsequent improvements in the plasma concentrations. And what was interesting about this lead too is that we also, it went hand in hand just by luck, I guess, with greater potency. So you can see here we're getting a quite strong inhibition of colony formation, even at 100 nanomolar. And you can see the DC50 here is about 6.7 nanomolar, slightly better than MS-21. And then this is our best compound right now, PG26349, which we found out, we started to first this summer, and we identified it. And as a very, very good degrader, its DC50 is 2.8, and we have basically 10 mg per kg injection leads to over like about 3 micromolar dose levels. And you can see here that here's the normal dose range that we've been testing. It's quite effective. We lowered the dose range so that this is again in the PC3 cell line model. And you can see now we were able to get it down to like where 10 nanomolar is giving us sort of equivalent growth inhibition that we see with capivasertib at 10 micromolar. So it's a pretty big shift in. Now, we still have a lot of work to do. We have to find out if there's off-target effects with these new molecules. There's plenty to do, but we're excited and we're about to put the molecule into mice. We made a batch that's sufficiently large to give to the mice orally, so we should be knowing in the next like month. By the end of the year, we should know if this is working in mice as we expect. We've done a pilot experiment with 2 mice and it showed efficacy, but we need to do an expanded experiment. So we have anyway developed a large series. So the summary of the first part of my talk, and I'm going to have to. I've been sort of slow and laborious here. I apologize, but at least thorough. But I'll have to sort of smash through the second part of the talk. But we've developed a large series of AKT degrader compounds. We've developed a first-in-class potent and selective small molecule AKT degrader that specifically degrades activated AKT. And then MS-21 is superior to AZD5363 for inhibiting the proliferation of PI3 kinase P10 pathway mutant cells in vitro and vivo. Aurora kinase B is a substrate of AKT and its downregulation contributes to MS-21 inhibition's effect. We saw that resistance to MS-21 in cancer cell lines containing KRAS and BRAF mutations, and it could be overcome by, in some cases, combining MEK inhibition with MS-21. Since publication, we have data showing that we can actually cause resistance by expressing KRAS or BRAF using a retroviral expression vectors in some cell lines. And then basically, I think just reiterating the obvious that if we could get a better therapy, for targeting AKT, it could be beneficial. I mean, I think the data here, from an optimist point of view, would argue that the AKT kinase inhibitor just doesn't inhibit well enough to lower Aurora kinase B. Right, and if that's really one of the beneficial benefits, and that we're seeing at least some output, and then obviously the feedback allows the cells to adapt so quickly and this feedback is really limited with the degraders because there's really hard for it to go through. If the protein's not there, the feedback is very hard for it to function. So anyway, I'm going to switch gears now and talk about P10 long delivery inhibition of tumor growth in mouse models, actually in syngeneic models. We published on this like over 10 years ago now, looking at tumor xenografts, human tumor xenografts in mice. But we've continued to sort of explore this area, both from a knockout point of view in mice and also overexpressing mice and also using AAV delivery. But back when we published our paper, we noticed we had an antibody that was specific for P10 lung that we made it and outsourced. And we identified this is a mouse GBM, and we could see very strong staining surrounding the tumor and histiocytes surrounding the tumor, macrophages surrounding the tumor. Back then, we didn't know anything about immunology in 2013, at least I didn't, to be very honest. And it was just sort of like this weird phenomenon that, and it just shows you how much that the cancer immunology field has matured in the last over a decade. And here's some breast cancer, not all the time, but 4 out of 50 tumors showed a very strong staining of macrophages in the tumor microenvironment. And this is just Western data from human GBMs showing that there's this P10 long is clearly upregulated compared to normal tissue, suggesting maybe the macrophages are the source of that there as well. So anyway, the first data we got is we made a knockout mouse and collaborated with a Columbia faculty member, Alice Prince, who works on asthma and also cystic fibrosis. And we shared the mice with her, and she was doing Pseudomonas aeruginosa infections of the lung, and identified that there were, here you can see, if you knock out P10 lung without affecting P10, you see more colony-forming units of Pseudomonas that survive after inoculation in lung material from the bronchiolar lavage, and also in the total lung itself. And then taking ex vivo macrophages, bone marrow-derived macrophages, and treating them with an infection of PAC or Pseudomonas aeruginosa, we could also see more colony-forming units in the knockout. So suggesting that the macrophages somehow were no longer functioning as we expected. So anyway, so just put that in your hat. Those are just sort of pieces of data, hopefully, that will start to come together as we finish the hour, and I will try to speak very quickly. So he generated, in addition, transgenic mice that overexpress P10 long using the an ATG conversion of the CTJ translation initiation codon, and putting them into a ROSA 26 locus and activating them at the 2-cell embryonic stage, and seeing how they affected the animal. And the first thing we saw was that, yes, we could see upregulation of expression in many different tissues. We could see actually measurable levels in blood plasma, which we documented before with human P10 long in our original paper. And we could see that the mice were quite small. By the way, this is not published yet. These mice are quite small, and this is very much reminiscent of AKT 1 and 2 knockout mice, which show very small size. And anyway, we see this difference in males and females. We see them and have the smaller size at the embryonic level. We also see that the overexpression of P10 long leads to reduced ability to lower a glucose in a glucose tolerance test, and also in an insulin tolerance test. Also, you can see here, this is just a muscle captured from mice at baseline, overexpressing P10 long or control, or in the presence of insulin. And this is in liver on the right. And you can see, the nice stimulation of AKT phosphorylation and its substrates and mTOR substrates, S6 kinase substrates. And you can see it's really diminished in the overexpression mice. So the reason we generated these mice was to see if they had an ability to affect the microenvironment for tumor growth. And so we had two, what we thought would be two potential syngeneic models. We ended up using MC38. It's a naturally P10 null colorectal cancer cell line that's syngeneic in C57 black with very low levels of expression of P10, non-detectable by Western, very low RNA level. And there's a paper reporting it to be mutated that characterized it. We transplanted both the MC38 into this, the P10 null, and we saw that tumors were smaller in size. This is a model FR1 that we made, which is it was originally, it's a PIK3CA mutant mammary tumor, originally a luminal an ER positive that now is converted to ER negative over passaging. And there we also see tumor growth reduction. We didn't see any difference in Ki67. We did see a subtle but measurable difference in increased caspase-3 in both models. And we could see by just immunostaining, you can see the clearly, this is the control, this is stained with P10, and you can see there's definitely some of the cells are much more positive than in the control, a little ambiguous using the classical immunohistochemistry approach. We also did this by immunofluorescence, which was much clearer. And again, you could see many of the tumor cells were expressing, had P10 long in them that we were transplanting that were P10 null. We also noticed, and this was kind of the first inkling in this model that the immune system might be involved, is that CD3 positive, there was an increase in CD3 positive cells in the tumor. that was treated in the tumor that had overexpressed PTEN long. And basically at that point, we have some other data on this model, but I don't have enough time to go into it, and I really should be done in 5 minutes. So we just switched to an AAV model where we thought we could get higher levels of higher titers of AAV PTEN long. And we did this in collaboration with Genentech. And so we basically, instead of using the classic PTEN long that we talked about before, we converted the secretion signal to another one. We tried light chain secretion signal, but we also used chymotrypsin B2s, or the human insulin peptide signal, and we tried them all out, and basically all of them expressed well in the liver. And then in terms of looking at secretion into the blood, they all looked about the same, but CB2 looked maybe a tad better, so we just started working with that one. And we noticed that if we we could see basically that IV injection of the chymotrypsin B2 PTEN long could lead to tumor xenograft regression. So basically, we implanted the tumors, we added AAV, and then we saw these are examples of mice where we had regressed tumors. Those are the most startling regressions. This is just more data demonstrating we can measure the serum levels of PTEN long. And also, again, I'm just going to fly through this. And this is just showing quantitation of a GFP control treated versus this PTEN long treated mice. The this you can see here that this is just comparing our transgenic mouse to the AAV, and then you can see that the level of expression is a lot higher in the liver tissue. And also you can see here that this is also looking at, we also made some phosphatase dead versions which express well and don't change phospho-AKT very much. And this just looking at a time course of this and of different wild type forms. Another mutant which is deleting the polyarginine sequence of PTEN long. And you can see the expression again in the liver starts to go down after 2 weeks. And this is another experiment where we used a log lower level of virus, and we could see still the benefit of the AAV vector, but now we're not seeing any complete regressions. And then this is just, I'll just flash through this because we're out of time. But yes, we could see PTEN long in the tumors when we did immunostaining. And this is just some other controls. And this was associated with lowering phospho-AKT and this CD3 phenotype that we'd seen earlier. And this is just another example of another tumor. And this is another example of another tumor. And this is a GFP control where we basically see no PTEN, phospho-AKT high, and low CD3. And we saw this, over and over again. This is a phosphatase dead, which didn't show any evidence of lowering phospho-AKT or recruitment of CD3 to the cells to the tumor. And this is another one that doesn't enter tumors, and we saw the same thing. So we did flow on CD45 positive cells. We saw a very dramatic change in the tumor microenvironment, where PTEN long lowered the number of total macrophages that we saw, increased the number of T cells as we would expect from the immunostaining. And then this is just looking at the flow analysis using an Aurora spectral flow cytometer. So we were able to look at many immune markers simultaneously, and we could see again a lowering in the number of dendritic cells, a lowering of the tumor-associated macrophages, however, increasing the TAMs that are type 1, increasing the number of T cells, increasing the number of CD8 T cells, and increasing the number of NKT cells. We're also seeing here nice evidence of somewhat more subtle changes in some of the other subtypes that were analyzed. Here is again the NKT cell broken down, and And you can see a change in PD-1 in the NKT cells as well. And then finally, the tumor-associated macrophages, we could see very nicely upregulation of CD80 in the tumor-associated macrophages, MHC class II expression in the and this is CD80 and MHC class II. And this was now in the TAM1 type, we're also seeing that same phenomenon. In the TAM2 type, less so, although we see some enrichment here. And there's just some more data, which I'm out of time. And then finally, single cell data analysis for the same CD45 population. Basically, one of the most predominant Go terms is antigen processing and presentation, and a variety of other antigen presenting stimuli. And you can see here, this is just looking at a series of genes associated with upregulation of antigen presentation related genes, and they're being upregulated in a variety of P10 long treated tumors compared to controls. And these are the different cell types, different types of tumor macrophages and these dendritic cells and regulatory dendritic cells, the TNK cells and the neutrophils, as well as the regular dendritic cells. So we're seeing this really quite robust immune awakening when we're treating the cells. And then finally, we can see by IF the macrophages in the tumor are positive for P10 long, co-staining with the macrophage markers F480. And we also could show that we could either, on the right, really robustly block the benefit of the treatment with an antibody to CD80, and for a brief period, partially block the benefit with the CD80 antibody. So anyway, so that's basically it. This is the group that did it. I'd like to just acknowledge Jia Zhu, who is the leader. He's now a faculty member at University of Alabama, shown here. He was a leader of both he's a remarkably talented scientist who is the primary leader in the laboratory for both the degrader project and also the P10 long project I just showed you, and also the other postdocs that are in the lab right now, and also, of course, the John Jin's lab, the Regeneron collaboration, and other faculty and collaborators at Mount Sinai. So thank you. Sorry, I went too long. Yeah. Hi.
And I guess one of the questions is, have you tried to use the P10 long and sensitize a resistant
We have not done that experiment yet. We're basically, I mean, it would make sense. It would work. We know that, I didn't really talk about it, but P10 is known to regulate PDL1 expression. And in fact, we see, I didn't show the data here, but we in fact do regulate PDL1 expression when we use AAV in the tumor. So they downregulate a PDL1 expression in the tumor. So we would expect to see a benefit from improving checkpoint activation, or dampening checkpoint activation, excuse me. But also, we haven't not directly tested the hypothesis using an anti-PD-1 combination. But it's a good idea. Yeah.
Where are you placing your bet? You get a really high quality, readily available.
Okay, so that's a really, so I think we are going to need to have a series of molecules. We can't bet on one molecule. What we do, though, need to have is good PK, right? We need good PK. So in a perfect world where we could get good PK on a series of molecules, I'd like to be able to have a series of molecules that have different kinetics for degradation. Because I feel like if you have a molecule that doesn't really turn the pathway off for 3 or 4 hours, it's going to give normal physiology. If you add insulin, it's like on, like acutely in a mouse, that signal's on for like 30 minutes, right? And it starts to come down really rapidly. So if we could get some kind of an understanding of the kinetics of degradation, because I think if we have something that's a really fast degrader, and I'm kind of worried about our best lead compounds, they could be too fast. They're going to be functioning just like a kinase inhibitor. Pardon? I think it could be really in a phase 1 program where you have patients with sort of very poor prognosis cancers, where they're willing to, and it's ethical to enroll them, but basically with a eye towards toxicity, because if we can see an improvement, if we can find a molecule that basically shows efficacy towards the tumor, and what's nice is we have these really clear biomarkers of on-target toxicity in the normal tissues that are really pretty common. Like rash is very common, diarrhea is very common, and obviously the glucose is just easy. We can measure that as a biomarker. So with relatively, I think, a small number of patients, we could figure out like how to sort of navigate the glucose toxicity, for instance, which could be a surrogate of the other toxicities, and then hope to find a window. I mean, yeah.
So my question really follows up on that, which is, do you have any data on lower toxicity, activated degrader?
Unfortunately not. We have identified a couple of, this lead compound is able to degrade AKT in mice, and we have another molecule that is not, which we can use IV, which degrades AKT in mice very well. So we can use both of those to sort of explore toxicity issues. Just to be clear, the original compound that we published is great because it doesn't, I mean, it's great. It doesn't degrade mouse AKT, right? So, we have a pure play there where we could dose it at really high levels and not see any toxicity. But, actually at that time, we did not know that. It wasn't when we published, but now it's very, very clear that it doesn't. So we're, I think, in a poised now to see. The other thing that we're doing, which I think is really interesting, is we're just trying to understand like how, obviously we can stimulate insulin to see if we can stimulate degradation, which would show that we can really regulate this process, but we can also block degradation with a PI3 kinase inhibitor because it's not going to be phosphorylated. So we're going to be playing with the space of seeing how we can modulate the cell so that degradation, so we understand under what context it's degraded. And we may find that there are degraders that are slightly different in their properties, kind of like the whole RAS field has exposed lots of interesting new space that we didn't know about until we had the molecules. So I'm relatively excited. I just think, we may run out of money before we can get the whole job done. But basically, I do think that there's definitely reason to think that this could be useful if we could just tackle this toxicity problem. Yeah. So, question in the back?
Yes. Just wondering, with these new degraders that you're testing out, have you looked to see if they are able to penetrate into the brain, or if they're in the brain? What would that mean for maintenance?
Yeah. I don't know right now. I think in general, the degraders don't really penetrate very well into the brain, as far as I understand. I'm hoping they don't, because the toxicity in the brain would be pretty severe. AKT3 is really a brain-specific protein, and we know that PI3 kinase inhibitors that have crossed the blood-brain barrier can be quite have a lot of toxicity, brain toxicity, particularly psychiatric effects. So I don't really know what's going to happen, but I'm hoping that we don't. I mean, obviously, we'd love to have a drug that's perfect and can cross the blood-brain barrier and treat GBM. I'm not saying, but I think I'm just, as a realist, trying to navigate all these different hurdles. I think it might be, at this point, something I'd hope the first generation we can just prove that they're actually better than the kinase inhibitor class. Ian?
Potential, but obviously focus on trying to get around the, on-target toxicity is what everybody's working on. what's your sense of the mutant allele specific PI3 kinase inhibitors as a way to do that? A lot of work.
I think they're great. I mean, actually, I think they could be the best. I come at this from a P10 perspective where they're not going to work for P10. And there's a lot of people with P10 disease. And I'm looking at it from that point of view. I think the rationale, if it was purely a PIK3CA mutant play, I would say that's the best way to go. But I like to go with the underdogs. Right. So, any other questions? Okay. Thank you.